Where do we draw the line in phylogeny? That is the question asked by critics of creation since the phylogeny challenge began. To answer that, we must look at many different components that not only show relation, but also what separates it. If you want to see if a car is similar to another, do you just stand back and look at the lot full of them and say, well, they all have tires and all have similar shapes, therefore that looks related enough for me? Or do you actually open the hood and investigate a little deeper? Obviously, you should not just stand back and look for similarity at something and saying, well, anything with tires was related, or anything living with hands must be related. That's generalizing, but that is exactly what secular evolutionists do internally when trying to prove common ancestry. They look at all the genes and say, look, anything that has more of these similar genes must be related. But that is a foolish way of doing it, because obviously we're going to have more similar genes with an animal that lives on land with similar body structure than we would to aquatic life. Here's an example for you. If we look at overall mtDNA between species, it is far too inconsistent to prove relation. They have discovered that mtDNA sequences between domestic dogs and the gray wolf differ by at most only 0.2%. That is to be compared with the huge 4% difference between some populations of the same gray wolf species. And if we look at the overall nucleotide sequencing, well, that doesn't determine relation either. We know this because some things have closer nucleotide sequences that are unrelated. We must dive deeper. Now, the big question. How much modern human variation is there in comparison with the overall animal kingdom? And how is the distribution of human genetic variation similar to or different from the broad swath of animals, especially primates? Well, DNA barcoding is that answer. It allows us to zoom in and address this very question for the very first time in history. When we look at DNA barcoding inside a single gene in mtDNA, we can see a clear genetic boundary in all animal life. Basically, DNA barcoding works through the comparison of nucleotide sequences in the DNA to that of the same gene in other species. This is one way we can differentiate and identify related taxonomically similar families of animal kinds, as the Bible says. As there are clear genetic boundaries we can see using this technique. You see, animals have two kinds of DNA. One we are the most familiar with is nuclear DNA. It is the genetic blueprint for each individual. But all animals have DNA in their mitochondria, and their mitochondria all contain 37 genes, and one of them is known as the CO1 gene. It is used to do DNA barcoding because it is the best for many reasons. Unlike the genes in nuclear DNA, all animals have the same set of mitochondrial DNA, proving a common basis for comparison. Canadian microbiologist Paul Herbert, who coined the term DNA barcode, figured out a way for us to identify lineage-specific relations by analyzing this gene. Original studies done in 2011 found how well it worked. Dr. Herbert believes that variation within a taxonomic family is restricted by an evolutionary mechanism called selective sweep. Sweeps occur because the CO1 gene functions are so important and constrained that they will always spread through all members of a related family to all future species of that family. The removal of this recorded genetic material of natural variability within animals is the critical feature that creates a distinctive genetic signature for each species tested that makes barcoding possible to test relation. Then, in 2018, they went testing high numbers of species never before done in history, and the results are now in. By analyzing the barcodes across hundreds of thousands of species, researchers found a telltale sign showing that all animals emerged at the same time as well as humans, and all have clear genetic boundaries from one another. So a dog and a wolf, though not considered the same species, both had low DNA barcoding gaps, showing that they are related to one another and on a family level, drawing a clear genetic boundary. While results also showed that a large DNA barcoding difference gap to all other tested animals that evolution says are related to wolves, like the brown bear, the sea otter, marten, fur seal, and badgers. All were distinctly different, having large barcoding differences. Now, the same was found in all humans, as when the human lineage was traced from modern-day humans to Neanderthal to Denisovan, results showed low barcoding gap differences, and when compared to the primate family, they showed large gaps. meaning humans taxonomically should be in a different family from primates. Here you can see for yourself the differences between life using DNA barcoding. Just look how similar butterfly species are to one another, yet how different they are compared to an owl. With CO1 sequencing, we can also see the difference between birds and bees, and relation can be mapped out easily. Now one more time, I want to cover selective sweep. Because the CO1 gene functions are so important and highly constrained, they'll always spread through all members of a related family and to all future new species in that family. So now, if primates shared a common ancestor with humans, then obviously this pre-human chimp hybrid ancestor would contain the base CO1 gene that would have been passed down to both lineages, meaning the CO1 gene would be just as similar in humans as it is in the chimp line because of selective sweep. In 2005, Vincent Savalanian stated that in order for any two organisms to be deemed the same species, they must share 88 to 98% of the genetic code that is chosen at the CO1 mitochondria gene fragment. This means that because chimps, which have over 60 base pair differences from us, they are unrelated. This is what related species look like using a heat map. Now, look at where the chimp falls in comparison to humans. Not even aligned at all. And there is no way around this new evidence. The germline always passes down the CO1 gene, meaning it's inherited without question, which is the reason why DNA barcoding works so well. The evidence is clear. A common ancestor to human and chimps does not exist. Now, as a critic, you might be thinking to yourself, but wait, there are similar species that share over 60 base pair differences, so how come they are still related but humans are not? Well, this comes down to percentage. I was only referring to primates when I mentioned the 60 base pair difference. The reason is different for other species for one main reason, and that reason is generation times. Since their mutation rates are nearly identical to ours, then all we need to look at is their generational time to figure out their nucleotide differences. So this makes it easy. Since they have their offspring between ages 3 and 6 years, then that is 3 times faster than humans or primates. So all you have to do is multiply 60 times 3, and your window of differences has now gone up to 180 differences, giving you your new window of what is related in the family. This can be done with anything. Now, combine DNA barcoding with taxonomically restrictive orphan genes, which show which animals aligned with one another on a family level. Then we add those two methods with a comparative statistical tool called heat mapping that catalogs the comparisons of all expressed proteins in a species because it provides visual representations of a group to which each species belongs to. Then you have yourself a trio of ways to prove and draw the line of where ancestry ends. Are there more? Yes, for example, zygote fertilization would be another example, but since it's not practical, we will not add these to the list. In conclusion, the largest genetic study to ever take place looked at hundreds of thousands of different species. They discovered a few things. Genetic diversity is about the same, proving that all life arose at the same time, including humans, and they all have clear genetic boundaries. This was so shocking for the scientists working on the project that they said, "This conclusion is very surprising, and I fought against it as hard as I could." They go on to say, "This study is one of the clearest, most data-rich, and general facts in all of evolution, that the existent population, no matter what its current size or similarity to the fossils of any age, has expounded from mitochondrial uniformity. If individuals are stars, then species are galaxies. They are compact clusters in the vastness of empty sequence space. The absence of in-between species is something that also perplexed Darwin." Now, be honest with yourself. What could possibly reduce all life on Earth to restart, even in lakes, rivers, and oceans? Only the biblical Noah's flood could do this. This was so shocking to the scientists on the study that even the logic behind these findings could not make sense to them. He stated, "All species show the same lack of barcode diversity. Although it is easy to imagine that humans passed through a bottleneck 170,000 years ago, it's hard to believe that the exact same thing happened in all species. Did herrings really pass through an equally recent bottleneck? Anchovies too? So this not only answers the phylogeny challenge, but it proves the biblical global flood as well.